Thank you, AJ. And I'm going to stay down here so I can see the slides as they come along. So good morning, everyone, and thank you for the invitation to speak here at K-State. You've got a geneticist and an economist. I'm not sure what you guys did to annoy the programming committee, but we'll do our best to be entertaining. And I really am here a little bit pinch hitting for an old grad student of mine who's a new faculty member here at K-State, Macy Mueller, who had a little boy 3 days ago, and so she asked if I would give this talk on her behalf. So I will do my best to make her proud. So the topic I was assigned was genetic modification in livestock. And genetic modification doesn't really have a definition, and often when people hear that expression, that's what comes to mind. The whole GMO debate and scary memes on the internet that if you eat GMOs, something bad will happen to you. And that narrative has been really hard to correct. And those of you that grow genetically engineered corn, like Roundup Ready or BT corn, know well what GMOs are, but the perception around them with the general public tends to be this very scary concept, and it's really around the technology of genetic engineering. But genetic modification, GMO, is actually, if you think about it, something that we do as breeders when we select animals. And so the reason that these dogs look different from their ancestor, the wolf, is because breeders of different breeds liked a particular phenotype or behavior, and they selected the dogs that displayed that characteristic. And the reason those animals had that characteristic was because of random genetic variation in the dog genome, or the wolf genome technically, and that then led to the different phenotypes or appearances that we see with dogs. And I've used this slide for many years, and I was thinking, well, surely it's not that hard to maybe do it with cattle. And actually, you can do kind of a similar slide with cattle if you want to think about it. We have an amazing diversity of breeds of cattle that have been basically genetically modified from the auroch, the originator of cattle, to the different breeds we have today. I did try and find on the internet if I could find one, and I think that was perhaps the best one I found, where an actual person really does resemble a cow, but that's perhaps not typical. But if you think about, we all started off in the origin of breeds, and breeders got together, formed breed associations and said, we want black cattle, we want horned cattle, we want shaggy cattle, whatever the breed definition is, and then that breed association moved forward with those, all based on naturally occurring genetic variation that exists in the genome of cattle. And so if we look, all animals are made up of DNA. Hopefully this is rudimentary fundamental stuff. I just want to make sure we're all on the same page. Every cell of an organism contains the entire genome. In the case of cattle, that's about 3 billion base pairs that makes up the cattle genome. And in 2009, we were able to get the entire sequence of the cattle genome, and we were able to then have a look at the genetic variation that exists between breeds. And I think it's important to realize that when we look at the DNA of cattle and we compare it to our DNA, we're actually about 83% identical to cattle because of our common origin, as in we all evolved from the same background. And so when we talk about transgenic DNA or DNA from other species, there's actually 83% that's identical between cows and people. So are we perhaps transgenic cows in a way? So I just want to understand that genetic variation, because when we sequenced the bovine genome and then did the 1,000 bull genome project, where we sequenced separately the genomic DNA from 1,000 different bulls, actually it was more than 1,000, it was 2,703, so it's a misnomer, the 1,000 bull project. But what we found was that there was literally millions of genetic variations, be they single nucleotide polymorphisms or SNPs, and little insertions and deletions that result in the amazing diversity that we see in our cattle populations. And sometimes you'll get a variation that causes a very big alteration in the appearance of an animal. And I give an example of double muscling, which is a naturally occurring mutation in the gene called myostatin. So myo is muscle, statin is stop. So if you mutate myostatin, you basically turn off the regulation of muscle and you get this very big hypertrophied muscle phenotype that you see in breeds like Belgian Blue. Not that conducive to calving when you've got a great big muscly calf that's having a difficult time getting out the birth canal, so often delivered via C-section. Not great for a production trait, but a very dramatic phenotype, nonetheless, due to one mutation in one gene. And if we look at the changes that breeders have been able to make over the years, we can actually make pretty big changes if we focus on one particular phenotype, in this case, height. And that's over the course of, 40 years or so. Was dramatic changes to the breeds. And I think actually the reason we kind of got more moderate after the 1980s was you couldn't actually see who won the show because the cattle were too big. And so I think we had to breed for cattle where at least you could see the head of the breeders. Otherwise, what's the point of having this picture, right? And so I think once we realized that maybe biggest is not optimal and had a more moderate breeding objective, we see something really interesting happening to the cattle population in the United States. So back in the 60s, there was an increase in cattle numbers. So blue is cattle numbers in this slide. And you see it went from about 90 million up to about 140 million there in 1975. And beef production, shown in red, kind of tracked along with cattle numbers. Then something really interesting happened. So we started to decrease cattle numbers, coming back down to about the 90 million that's true now. But what happened there was we produced more beef with less animals. That is the definition of sustainability. Well, how do we do that? More beef per animal. It's the only way you can do that. It's the only way that slide makes sense. And that was when we started to use well-informed breeding objectives that selected for optimal animals rather than perhaps maximum animals that we might have seen in the 80s. So that's a little bit of a history of conventional breeding. I'm now going to move to biotechnology or genetic engineering, and I hope this audience is aware of what that technology means, and I would assume, because many people grow genetically engineered corn, that perhaps that's common knowledge. It's grown by literally millions of farmers throughout the world, but we haven't really been able to use it in animal production. And that's a little bit due to the pushback against genetically engineered plants, but also the difficulty of getting these products through the regulatory process. It's very expensive and needs deep pockets like Monsanto to bring the products through, and we just haven't had analogous interest from the animal breeding industries. So what we see is back in 1985 was the first transgenic livestock that were ever produced, so 40 years ago now. And transgenic is defined as having DNA from a non-sexually compatible organism. So basically a DNA construct that gives your animal a particular characteristic, maybe disease resistance, depends what the construct is. But when that DNA doesn't come from the species that you're working in, that's the definition of transgenic. And so, for example, BT corn has DNA from a bacteria that results in it having its BT characteristic. So it's transgenic. We call it genetically engineered because, to me, transgenic's a little bit of a confusing term because, as I said, we share 83% of our DNA with a cow. So is it foreign? If you bring a human gene that's identical to a cow gene into a cow, it's no different. And so, to me, it's all a little bit muddy, but that's basically what a transgenic is. And we see, perhaps if you worked in this field, you might recognize some of the animals in this slide. So you can see the fish there, that's the Aqua Advantage salmon, which is a transgenic fish that gets to market weight in about half the time as a result of having a construct from the Pacific salmon that basically lets it grow more consistently to get to market weight. You'll probably recognize Dolly, the sheep. She was the first animal that was cloned from an adult somatic cell cloning. Very dramatic when it happened, but it's now used fairly routinely in industry, and many of the elite bulls in the AI sector are derived from clones, and that's the technology that's used a fair bit. But the real thing that I'm going to talk about today happened around about a decade ago when we had the introduction of what's called gene editing. And so gene editing is different to genetic engineering. So genetic engineering, again, is introducing a transgenic construct into an animal or a plant that gives it a particular characteristic you want. Gene editing is basically using a technology where you can tell a nuclease where to go make a double-stranded cut in the 3 billion base pairs that occupy the bovine genome, and you can inactivate genes that way. So you can turn off specific genes, like, for example, myostatin, if you wanted to get double muscling, or perhaps a gene that makes an animal susceptible to disease. And so it's the ability to go in and tweak the genome in a very targeted way, which is kind of what breeders are doing, but just based on naturally occurring variation. In this case, we go in and we say, you know what, I really want the polled allele in my Wagyu. I'm going to go in and basically introduce that and turn off horns so that I get polled Wagyu. So you can go in and alter the endogenous DNA of an animal is basically what it is. So the reason that we were excited when gene editing came along is, as I said, because of the pushback on genetic engineering, there's only ever been 2 approved products globally for food production in animals, and that is the Aqua Advantage salmon and a small application for a knockout pig that basically doesn't express alpha-gal, and so people that have the red meat allergy that's carried by the Lone Star tick that are allergic to red meat theoretically could eat this pig because it doesn't have that particular allergen in it. But that's a pretty sad legacy after 40 years, 2 products, versus plants have millions of farmers growing it all over the world. So that blocked us from using the technology. And with gene editing, because we weren't introducing a transgenic construct, to me, I was like, well, this is basically just conventional breeding, but done more intelligently. That's not going to be treated the same as what genetic engineering is from a regulatory perspective and hopefully from a public perception perspective as well. So I am going to try to explain genome editing. And so often when you hear GMOs or the discussion of GMOs, there's, for whatever reason, some random picture of a tomato getting an insulin syringe poked into it. This is not how we do genetic engineering in animals. That's not how we do it in plants, but it's a very emotive image that captures the fear-based narrative around the anti-GMO movement. I'm going to try and explain gene editing to you without having to invoke a tomato with an insulin syringe in it. So basically, this little slide is a summary of what we're talking about. So on the left there are the different editors, and basically they're a very fancy pair of scissors. They can be programmed to go to any location in the 3 billion base pairs in the genome and make what's called a double-stranded cut, a nuclease-induced double-stranded break. The cell sees that double-stranded break and it tries to repair it because it does not like its chromosomes being cut in half. And there's naturally occurring repair mechanisms that are involved in just normal cell division that will come in and repair it. And if it repairs in a way, and I'm going to call it the left-hand pathway there, the SDN1 pathway, where a small, maybe you drop a base pair or you add a base pair such that you no longer have the protein in the right reading frame, you can inactivate a gene. So you basically knock it out, is what it's called. And that can be useful if you're targeting a gene that creates something you don't want. For example, susceptibility to disease. Alternatively, you can go the right-hand pathway there, which is the knock-in pathway, where you provide some template, and here it's called donor template. It's the blue-colored image. And that will integrate into the double-stranded break and through homology-directed repair, and you can introduce a useful allele. For example, the polled allele from Angus could be introduced into Holstein genetics to give you polled Holstein. And that's what the right-hand pathway is. So either knocking out a gene or introducing an allele from a different breed. So maybe if you want polled Holsteins, you probably don't want to cross it with an Angus to get the polled characteristic because you've now got half Angus, half Holstein. You want the elite genetics of the Holstein for the dairy industry, but you just want that particular allele coming in from the beef industry. So that's my explanation. I'm a big advocate for movies and pictures tell 1,000 words, and so I'm going to play a little video that not only does it better, but also has an American accent to make it easier to understand. So let's see how we go.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting unnaturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual, what they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has two components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
Okay, so not one insulin syringe in that whole video. So how do you go about editing a cow? How do you do that? So basically, there's two different ways that you can approach it. And at the end of the day, what we want is to have an edited cow that has both alleles, that typically you want both alleles to be converted to the desired allele. So for example, you would, like in the case of horned animals, if you're trying to get them to be polled, you want both the mum's and dad's allele to be changed to be the polled allele. So you want to get what's called a homozygous non-mosaic animal, and that's indicated in this image with a little purple cow that has two pluses, so both alleles have been edited. That is our ultimate aim, and what's most important is it has to be germline edited, such that the semen and eggs from the resulting animal pass that on to the next generation, because as breeders, if we haven't done that, we haven't done anything. And so this is quite different to human somatic therapy, where you're trying to treat like a patient with sickle cell. We are trying to do germline editing, and so we have to get to the germline somehow. And there's two basic ways that we can do that. So the first, shown here, is through cloning. And so if we start with the purple cow there, she's going to be the oocyte donor. We actually don't care too much about her genetics because we just want the egg. And we're going to remove the nucleus from that egg and produce an enucleated oocyte. And then we're going to have a cell dish where we're working with cells from an elite animal that we want to edit, and maybe we're going to knock in the polled allele, for example. We'll get the edit we want. And then if you remember, I guess, oh, that does kind of work. If you remember from Dolly, basically we're using Dolly technology here to then transfer a 7-day blastocyst in like normal embryo transfer. And 9 months later, we will get a calf that is exactly replicating the cell that we cloned. So that is cloning. Cloning's great, except it's not. So it's great in that you get germline transmission, and for sure your animal is going to be an exact copy of the cell you put in there, but you get a very low rate of efficiency, as in it's high degree of clones don't make it because they don't get reprogrammed correctly. And even after, 30 years, we really haven't got that sorted yet. So some groups like Transova do a lot of cloning and have got it a little bit optimized, but it's pretty low rates of efficiency. Also, you're kind of editing the last generation. So you're working on a cell line that derived from an animal, and somehow you know that animal's good. Maybe he's got progeny testing and he's been around a few years. What you really want to do is edit the next generation, because breeders are always, every generation should be getting better and better. So the other way to approach this is to actually pick up an oocyte from an elite donor, so this would be the germplasm that you actually want, and then you will fertilize that with sperm from an elite male to produce the next generation of amazing animals, and then you can edit it in the zygote. So after the sperm gets into the egg, you edit the zygote, and then it goes into embryo transfer, just like with cloning. And then if you're really, really lucky, what will happen is you will get a homozygous non-mosaic animal first off, and that's indicated there with the little purple homozygous non-mosaic. Or you might get heterozygous non-mosaic, meaning that one allele, so maybe dad's allele got edited, but the mum's allele didn't. But what happens more commonly, and I can tell you this because my lab works in this, is you get mosaics. And that's indicated here by the animals that have multiple colors. And a mosaic just means an animal that has more than one genotype in it. So what happens there is you'll get some cells that are edited and some cells that aren't. And hopefully your edited cells go to the germline, but maybe they don't. Maybe that animal has just normal semen and the edited cells are in the rest of the body. And so you basically then have to breed that animal to get it to be ultimately heterozygous non-mosaic and then get it all the way to homozygous non-mosaic. Well, cow generation intervals, what, 3 years if you're lucky? So that's a long process, and so there's disadvantages of using microinjection of the editing reagents into a zygote is that you often get these mosaic animals. You can get pretty high rates of knockouts. So we are routinely getting homozygous knockouts using this technology, but it's really hard to get the knock-ins where you're adding that template and trying to get an allele, for example, from one breed into another. So they have their pros and cons, but that's kind of how you go about editing a cow. And so those are your options. Why? Who cares about how? Why? Why are we doing this? Well, we haven't really changed our breeding objectives just because we've got a new technology. So what we're trying to do is still pursue our breeding objectives, but now we have a method to bring in a useful characteristic in a way that's very targeted rather than having to use breeding. This is a review that we did last year looking at what people were using genome editing for around the world in the peer-reviewed literature. And this is broken down by ruminants. So blue is ruminants, cattle and sheep. And then the monogastric mammals are purple, the fish are orange, chickens are yellow, and insects are red. And so not surprisingly, a lot of people are targeting yield. That tends to be an important objective of most breeding objectives. A lot working on reproduction, and I'll get a little bit into why that's an interesting aspect, but things like producing single-sex offspring for chickens, for the layer industry. We don't want male layers, they don't produce eggs, and so they get killed at birth, or hatching actually, and that's literally billions of chickens that go that way. If we could only produce females, that would be much more efficient. Maybe you've got a terminal sire, you'd like him to just produce males because you really don't want heifers for terminal purposes. So there are reasons why you would want that. Biotic stress refers to disease resistance, and that's a huge trait that's being targeted. Producing hypoallergenic products, multiple things. Color, I'll get to that in a minute. And then things like polled that affect animal welfare, for example. So that's kind of what it looks like. So let's have a look at what types of things are being done in cattle specifically. So you might want to knock out allergens such as the alpha-gal that gives you the, if you have red meat allergies, that's what causes it. You might want to give animals more heat adaptation, so that's the SLIC allele, that's a prolactin receptor knockout that results in the SLIC phenotype that you see, for example, in Senepal cattle, keeps the body temperature about 1 degree lower than non-SLIC or conventional animals. Sex ratio skew I mentioned a minute ago, unwanted development like boar taint. If you don't want your male pigs developing boar taint, you can knock out the genes that result in the production of that. Increased yield, myostatin, as I said, probably not great for commercial cattle producers in America. We don't routinely C-section our females. Disease susceptibility PRRS virus, that's the first product probably going to come through the approval process in America. That is a pig knockout for porcine reproductive respiratory syndrome virus. That is a devastating disease. If you don't know anything about pigs, it just rips them apart and rips families apart when their herds get it too. And then surrogate germline I will talk about in a minute because that was one of the projects that Macy was involved with when she was a student with me. So these are some examples of different countries, different applications that have actually been reduced to practice, meaning there's animals that exist with this characteristic. It doesn't mean they're commercialized yet. So, for example, in New Zealand, they have knocked out beta-lactoglobulin, one of the main hypoallergenic aspect or things that make you allergic to milk. This is an example of the SLIC mutation, again, as evidenced in Senapol, where you have this slick coat that keeps body temperature about 1 degree lower than conventional animals. This one's an older one now. I think this was 2015, so we're, a decade into this, where they were able to inactivate myostatin in Nelore breed cattle, and the bull on the left, you can see, has more muscle, but it's not as dramatic as the phenotype that you see with the double muscle gene. And that's one of the advantages of genome editing, is you can be very targeted and specific about which allele you bring in to maybe not have such an extreme phenotype as you see in the double muscled animals, but maybe you put 15% more yield on them that doesn't interfere with birthing and all of the things that are associated with double muscled animals. And so yield is still an important trait, and I sometimes have people say we shouldn't work at all on yield. It's still what drives the profit. And so I think things like that potentially do have some utility, especially in animals like Nelore, which don't have a very high yield if you compare them to conventional kind of English type cattle. This is another example from New Zealand. This is an interesting one. So I mentioned coat color. So they altered an allele in cows that makes black and white Holsteins into, I don't know what color I want to call that, light beige Holsteins, with the idea of not absorbing so much heat and to try to adapt them to a warming climate. So conceptually, you could do the same thing, for example, in Angus and make them whatever color you want to call that, beige, I guess. And then this is the germline complementation, which I think is such an interesting idea. So it's not editing a gene per se, but it's editing in a way to alter breeding program design. So what you could do in this case, so say you want your commercial sire to be a Brahman-influenced animal, so example in the south of America, but you don't really want the genetics from a Brahman passed along. You would like him to pass along genetics, let's just say from an Angus animal. So you want the heat tolerance and all of the attributes that come with that, with the indicus characteristics, but you'd like his germline or his semen to be derived from a different animal that's got maybe not well adapted to that environment, but it's the animal you'd like to use. So what we can do is use gene editing to knock out the germline of that host. So he is no longer fertile, and now we're going to take some cells from a donor, and we're going to complement that germline with the cells from that donor, such that this animal now has blue balls in this example. And then when he's put out naturally service mating, he is going to produce Angus offspring, so basically derived from those donors. And if you think about that, why would you do that? Well, it's hard to AI, artificially inseminate, just to be clear, commercial cattle, especially in large ranches and Northern Australia, for example. And so typically what we do, if we want to do that, is we have the elite seed stock sector, then we've got the multipliers, and then we buy commercial bulls. They go out, and the natural service mating is how that gets to the final animal. And there's this genetic lag between the seed stock sector in terms of their genetic merit and the commercial sector. So one way to get around that is to basically use AI. Another way would be potentially to use a surrogate sire to deliver AI quality semen through natural service. And to me, that application has some real beauty for the extensive pastoral industries like sheep and cattle, where it's difficult to implement AI. So that's one example that's going to be coming. I think my clicker is, oh, there we go. So that's knockouts. What might we knock in? Well, again, we haven't really changed our breeding objectives. We're just using a different technology. So disease susceptibility, unwanted development like horns, thermotolerance, improved food quality and nutrition, and again, the sex ratio skew. So what are some examples that have been reduced to practice here? I don't think we're working anymore with our clicker. Okay, you're gonna be my clicker. I'm gonna point to you when it's clicking time, okay? Here we go. All right. So here's an example from China where they've done a knock-in to produce tuberculosis-resistant cattle. Here is an example actually from quite close by. So this is work that was done at the Meat Animal Research Center, and I think the animal's actually housed at Nebraska. Basically, what they did here was they hypothesized that if they changed a small section of the gene called CD46. They basically switched out 18 base pairs, so there's 3 billion. They switched out 18 and changed the 6 amino acids in this protein. They hypothesized if they did that, that would produce cattle that are resistant to BVD, bovine viral diarrhea virus. And they did it in a gear. And the gear is that top left animal there that's looking at us with the long floppy ears. That's what gears normally look like. That's not what a gene-edited animal looks like. It's just what a gear looks like. In this case, it's got that switch that's happened, so it's called the ALPTFS animal. And that animal, when exposed to BVD, was not susceptible to that virus. So it's a virus-resistant animal in this example. So that was done by using a knock-in of that 18 base pair region. This one, now it's working again. This one is actually one that I was involved with, and so this is the polled example. And for those of you that don't know too much about polled, and if you're Angus breeders, you're probably thinking, well, why not just use an Angus? And you've got a good point there. But in terms of, Wagyu, for example, is not allowed to crossbreed their full bloods, and dairy cattle don't want to bring in Angus genetics. So in this case, you can go in and target it and alter the allele. And so basically, for those of you that are interested, the gene called polled has a recessive trait that is growing horns. So in other words, the recessive trait grows horns. There was a naturally occurring mutation that occurred in Angus or Celtic beef cattle many moons ago, where basically a 10 base pair sequence in the gene was replaced by a 212 base pair insertion. So this is nature genetically engineering cows to produce cows that don't grow horns. And so what was done in this example is this bull had that allele brought in, and it's in both mum and dad, so both alleles, and we crossed that with horned Herefords at UC Davis. So again, horns is recessive. We've got a dominant bull, and so we're going to get heterozygous offspring with one allele coming from that edited bull and one allele coming from the horned Hereford mother. And so what did those calves look like? Not that, I can tell you that much, because it's a dominant trait, and so they basically were just normal calves that didn't grow horns. And so this is reducing it to practice in concept. And this is a particularly important trait because some countries are getting ready to not allow the use of horn genetics in their production systems. And Australia would be an example of that. You're not allowed to ship anything out of Australia with horns. And there's a big push to not allow animals to get used that have horns. That has really big breed implications for Wagyu, for example, where they have horns and they are used a lot in those northern cattle. And so if you could introduce that trait into those bulls, that would be a very important trait. So we did analyze the 6 offspring that we produced that didn't grow horns, and we phenotyped them for 2 years, and they didn't grow horns the whole 2 years. Shocked. Not a very exciting experiment, but one that had to be done irrespective. And so what you can see here is the animals with horns were produced by a normal Holstein bull. The animals without horns came from that genome-edited bull. And we had 5 males that were born and the 2 females. So the one on the right there, her name is Princess. She was the offspring of the gene-edited male. And then Cinderella was right beside her there. And you can see that they didn't grow horns. And interestingly, there's some work being done out of Canada asking the public what they think about using genome editing for removing horns. And actually, in conclusion, most participants were positively predisposed towards using GM-polled cattle. And so that suggested that there is an interest in doing something that has a benefit to the animal, or a welfare characteristic in this particular case. And we have done a lot of outreach, it's kind of my job, I'm extension, with the public on this, and have had engaged quite a lot of press in it. And so we were approached by the Wired magazine to get a cover shot of Princess for the front cover of their magazine. And she came in and she actually was looking a little bit rough. And so actually Macy, who was a good show fitter, cleaned her up for her cover shot, because it's important to look good for your day in the sun. And so here they are. They came in with 4 people to take a picture of a cow. They had a photographer and an assistant and a director and another assistant. So much to the amusement of our feedlot. But you can see her there with the black in the background. They actually wanted to run her in a green room on campus, and that got a big no from the green room operator. Anyway, there she is, and we were quite happy because we had her looking pretty, and nothing could go wrong, right? Oh my God, that is nothing like what she looked like on that day, but she's got kind of this sinister look about her. It's like, it does look a little bit like she's a little demonic or evil in some way, and I felt bad for her because she was a good girl. And then I saw the picture they printed of me, and I'm like, oh my God, it's like, you know, don't get on my wrong side, apparently. But the media always kind of do this, like just want to create a little bit of drama around it, and that's kind of annoying. So where is this stuff happening? This is a little bit confronting. So this is that previous slide I showed what's getting done in which species. This is where that research is getting done. And you can see there's one country there that's got well over half of all applications. That tells me China is going to be using this technology in their agricultural production systems. US is second, and then you get a little smattering of other countries. And I'm going to just touch on regulations because I think it's such an important issue, and it's actually something that potentially will block it in the United States at the moment, because I don't think we have a very fit-for-purpose regulatory approach as it affects this technology. So I've talked about natural mutations at the top there in green, and that's not regulated any differently to conventional breeding. So that is conventional breeding when you think about it. So those products can go to market. They have to be safe. All food has to be safe. That's the US law. It doesn't matter how you produced it. But there's no additional regulations you have to go through if you breed a new breed of cattle, for example. Then there's going to be the changes that are possible with genome editing. So the small insertions, deletions, that left-hand pathway, using a template that comes from another breed, for example. That could be done using conventional breeding, but this does it in a very targeted way. And then there's kind of a red line at the bottom there, and then there's recombinant DNA insertion, and that's the transgenic kind of technology. That one has this really arduous regulatory evaluation that's multi-generational, that costs literally tens of millions of dollars, and has really not been something that the animal industries have been prepared to do. So where to draw the line is where the countries around the world are wrestling at the moment. At the moment, the line has kind of been drawn where that green dashed line is. So if you are working with an application that has a transgenic sequence, so just a reminder, transgenic different species in your species, so a non-sexually compatible species, then you're going to be regulated as a GMO or a foreign DNA. And then, okay, if you could have created it using conventional breeding and there's no foreign DNA, then you're going to be treated as conventional. And that's kind of the approach that many countries have taken, and I'll explain how that's happening. Okay. All right, so this is basically the regulatory landscape as it relates to genome editing. The green, which is really highly evident in South America, is basically saying if there's no transgenic DNA your conventional breeding. You don't have to do additional GMO regulation. You can go to market. Then there's countries, oops, back. Okay, yep, back, yep. Leave the map up, please. There we go, nope. Okay, then there's countries like South Africa there who say, we're going to regulate everything as a GMO. We don't care if it's a single base pair nucleotide change. Even though there's millions in cattle, we're going to regulate it as a GMO. So that's the most extreme. Europe's thinking about going that way. They're still making up their mind. But you'll notice that America's kind of hashed there. And I'll get to what's going on there. And it's important because where the regulations are kind of sensible, this technology is taking off. Where the regulations are basically cost prohibitive, this technology is struggling. And so that's kind of where we are. Okay, now you can go to the next slide. All right. Oop. All right, Argentina was the first one out of the gate with this. They basically say, no transgenic DNA, no foreign DNA, you're good to go. And so they have actually given what's called the regulatory nod, or non-GMO determination, to several applications. For example, slick cattle, Red Angus in this case, that were put through by Recombinetics, have been given the okay to go into the marketplace if breeders chose to do that. They've also given the okay for a polled, slick Holstein to go to market. And basically, if there's no foreign DNA, no novel combination of genetic material, you're non-GMO. Okay, next one. Next slide. Okay. And then it's similarly in Brazil, which has mimicked very much what Argentina's doing. The semen from the bull, the Nelore bull that I showed earlier, which has a knockout of that myostatin gene, good to go to market. The male that has SLIC, so the prolactin receptor knockout that mimics the SLIC phenotype that you see in Senepal cattle, good to go. Both considered non-GMO in 2021. So both of those South American countries could technically introduce these edits into their cattle and put those products on the market. I won't talk about trade just yet because that's another issue. But then, so interestingly, the SLIC has been given non-GMO determination in those countries. Okay, let's get to the United States. So the FDA came out with a guidance last year that has broken down gene-edited animals into 3 categories. So the FDA continues to consider that all gene-edited animals are unapproved animal drugs. It is the most ludicrous interpretation of drug I've ever heard of, because genetic variation is not a drug, because we all genetically vary from each other. Does that make us unapproved animal drugs? You know, it just is a silly thing. So they've said there's a bunch of genome-edited animals that we're not going to pay any attention to, even though they have an intentional genomic alteration. And I put up there the example, you may have seen in the news this week, if you go in and genome edit a mouse to have a woolly mammoth-looking coat, that's not going to be regulated by the FDA because it's a mouse and we're not eating it. So none of our food animals are going to fit into category 1. So let's have a look at category 2. Next one, please. Okay, products for which we don't expect developers to submit an application for approval, but we want them to submit data to enable us to determine if there's any unique risks for this product. And what they're interpreting that to mean is if a mutation already exists in our food supply, so for example, the SLIC cattle, Senepal, we already eat that. There's no food safety concerns associated with that. In the case that we're talking about here, the animals had the SLIC gene edited, they had the SLIC phenotype, nothing else about them was different, they just had that. In that particular example, they used this category 2 to classify 2 animals, next slide please, that were produced by Recombinetics or Excelligen in 2022 to be what's called given enforcement discretion, which is a really wonky term for when a regulatory agency makes a determination that in this case, you are selling an unapproved drug, but we're not going to exercise our right to enforce the law because it's low risk. Now, I don't know if that gives you a level of comfort. It does not give me a high level of comfort because, I don't know, administrations could change and so could the decision to exercise enforcement discretion. And then if you've got 100 cows out there with a slick allele, you might suddenly have 100 unapproved animal drugs running around on your property because that enforcement discretion was revoked and they are now going to enforce the law. But that's what's being done for products like that. And then the third category, next slide please, is products where basically it's a transgenic product or a product where we haven't routinely eaten that in our food supply before. That has to go through the whole GMO approval, which again is multi-year, multi-million-dollar regulatory approval. Not something you probably want to get into if you're developing things. So a lot of companies are working in that space in category 2, where you're just basically not introducing any transgenic DNA. So that's where we are in America. Next slide, please. And so this slide summarizes all of the gene-edited animals that have been given a regulatory determination anywhere in the world. And what you see is that the South American countries, so Argentina and Brazil are up the top, and they're starting to get a list of a few different animals that have been judged to be non-GMO that could go into the food supply. So we have, for example, heat tolerance with the slick animals in beef cattle in Argentina, dairy cattle with the polled heat tolerance, cattle with myostatin in Argentina. And then you'll see in Brazil, similar portfolio, but also, importantly, the pig with the CD163 mutation, that's the PRRS-resistant pigs, have been given a positive regulatory determination in both Colombia and Brazil. Not yet here, because they're going through the full approval process with the FDA, and they're hoping to get approval this year, 2025. Interestingly, Japan's there. Japan has been historically very toey about this technology. There were some fish that were produced by an academic researcher. Actually, one of them's a myostatin knockout fish. Fish that get to market in less time, eat less feed, good for the producers. And basically, the university professor that developed these fish said, hey, you know, it'd be really fun. I'm going to just form a little company and sell these fish. So he went to the regulators and he said, here's my fish. Are they GMO? And they said, no. And he said, great. Took them to market and they're for sale to the public in Japan. That's how it's done. You know, we're down the bottom there with 2 animals that have enforcement discretion for slick, period. Potentially the pig this year. We're not going to keep competitive if we're not allowed to use technologies that our competitors are able to use. Next slide, please. And now we get into the fun bit. And this is very relevant for this audience because not only do you need regulatory approval, you need to work with your breed associations. And so the breed associations are being faced with this, what the heck are we going to do with this new technology? And they're rightly cautious and nervous about the GMO debate and the pushback from the public and RBST. And there's a history of misinformation around biotechnology that makes them justifiably nervous. So let's have a look at what the breed associations are saying. So here is the Angus Association, and if you want to go and read the whole big blurb, it's at that URL. But basically, each specific IGA, that's intentional genomic alteration, is the FDA's terminology, will be based on its ability to provide sufficient benefit to advance the Angus breed. And so that breed association is saying, show us why we would want this in our animals. So for example, maybe someone wanted to bring slick into Angus genetics, and actually that's what happened. The breed association in this case said they've reviewed an application for basically a PRL slick IGA, so that's just shorthand for introducing that knockout into Angus. And at this time, we are not going to approve that animal for registration because there's too many unanswered questions to confidently assess the risks and benefits to the Angus breed. Next slide, please. This is the Hereford Association. So they also are going to look at the impact of introducing that gene into the Hereford population. And interestingly, eligibility for registration is restricted to those animals that have been reviewed, and this is important, and approved by the applicable regulatory agency. I just went through a bit of an explanation how we're going to have this enforcement discretion probably for most of the traits we're introducing. Is that an approval? It's not really. It's enforcement. It's not category 3. It's category 2. So is that going to run afoul of how they're being regulated? I don't know. It's hard to say. And I don't envy the breed associations. They're trying to work with the best information they have. But the wording here potentially runs afoul of the US. Probably fine in all of the other countries, but the way the US is wording it, that's not going to be an approval. Next slide, please. And here's Wagyu's approach, and they also reserve the right to deny or cancel registration if they don't think that edit is going to advance the Wagyu breed. And so the breeds are looking after their reputations and wanting to see that it's going to benefit their breeders, as breed associations do. Like, that's how we got these breeds in the first place, and they are the ones that should be making these decisions. And they have to be approved by the regulatory agency. Again, that language potentially runs afoul of the way it's being approached in the United States. Next slide, please. Oops, previous slide, please. Okay, the Red Angus has agreed to accept both heat tolerance, so slick, and coat colour traits, pending, and this wording is good, final regulatory decisions from the FDA, which that could include the enforcement discretion approach, and Nexelligent is currently working to get those enforcement discretions for those 2 traits. So this would be Red Angus that have the slick allele, and then this next slide is kind of an interesting example. So this is from next slide, please. Is from the R.A. Brown Ranch, and this is from a Facebook post, if you can believe, back in September 2021. And what was done here is basically a Black Angus animal was edited to introduce the red allele, which is normally recessive to black, introduced the red allele in the homozygous condition, and then altered the genome at a different allele that makes red dominant. And that allele came from red Holsteins. So they made what was a Black Angus into a Red Angus that now will breed true to red because it's got a dominant red in its genome. And that's what that calf is there. And so far hasn't, to my knowledge, got regulatory enforcement discretion. But you can actually alter defining attributes of a breed in this particular case. Why would you want to do that? Well, maybe you want to expand the genetics base of red Angus, and you want to do it right away without having to grade up. And so you can basically, in one generation, make a red Black Angus, for want of a better expression. Okay, next slide. What are the dairy people doing? Well, they're a little bit ahead of you guys in terms of the beef industry because they've been dealing with this for a longer time. But they have said that they'll allow the registration and they define what genome editing is. And then the registration name of the genome edited animal will include the suffix GE. That is the acronym that we have used for genetic engineering for 30 years. That is not the acronym that we want to use for this technology that we are trying desperately to differentiate from this very controversial technology. That's not going to help very much. But anyway, that's what they've done. Holstein took it up an extra notch. Next slide, or next click, please. Oop, previous one. There we go. And they did something really interesting. So they said, if you edit and bring in an allele from another Holstein, then we're going to treat that differently because it's going to be called an intrabreed Holstein versus if you bring in an allele from Angus, for example. So even though they're sexually compatible, we're going to differentiate animals that have an allele from a different breed. So that's a genome edited non-Holstein genetics. And then the last one is basically if it's not sexually compatible. So that's a true transgenic animal or genetically engineered animal. So they've taken it a little bit further, and then the World Holstein Friesian Federation, because we've just been talking US breed associations, there are breed associations in every country, and then there's also global breed associations. So the World Holstein Friesian Federation actually, I thought, did a pretty good job of explaining what genome editing is, how it might be used, and they are recommending that their members use the term cisgenics for when the genome editing involves DNA within the species, and then transgenics for when it involves DNA from outside the species. And that to me is a really good way to do it, because that kind of aligns with how the regulatory frameworks are lining up. So if it's transgenic, it's treated as a GMO and differently to if it's not transgenic, whether it's just a knockout or maybe an allele from a different breed. And so that's kind of where we're at with the different breed associations. Let me see if I can get this to work. Nope, next one. So how do I envision gene editing might impact the breed associations in the future of genetic evaluation? Well, I think if you wanted to, you could go in and repair genetic defects. So if you had a very elite bull that's a carrier of a genetic defect, you could go in and fix that. You know, another way to do that would just be to not breed carriers. That's kind of how we manage it at the moment, but you could go in and do that. And sometimes with very narrow breed bases like Wagyu, where you're not allowed to crossbreed with anything, and there are some genetic conditions there because they're super inbred, you might go in and do that for a very valuable bull. Most importantly to me, it offers an opportunity to introduce useful alleles from one breed to another. And so you can bring in polled into Wagyu genetics. That's to me, the power of this technology. It's currently, and I think for the foreseeable future, will be used for simple traits. That is Mendelian traits where one gene affects the characteristic. So things like horns, slick results in that slick phenotype. We're not going to be able to edit them to have polygenic traits like marbling, for example, unless we find a trait that has a gene that has a really big effect on that. And there hasn't been that many big effect genes found, despite the millions of dollars we've invested in all of the genome-wide association studies and what have you. So I think it's mostly going to be used for primarily Mendelian traits. And it could potentially be used, as I said, to alter the defining characteristic of a breed. And that's why I think it's so important for the breed associations to be making these decisions, because if having highland cattle have horns is absolutely core to your being, you don't want to introduce the poll trait because that has now altered your breed. So I think those are the types of discussions that the breed associations are dealing with. If you're interested, there's guidelines at the Beef Improvement Federation, which is a super important resource as to how to, what this technology is about, how to register mosaics. So for example, those 2 slick animals that were given enforcement discretion, they are not, they're not going to breed true. They're mosaic. They have some wild type DNA left. In other words, they could still pass on the non-slick genotype, even though they have been edited for slick, because not all of their cells got that edit. So that affects, and that's true if you buy a heterozygous bull, right? Half of his offspring are going to get one and half will get the other. So it's not new, but you need to be aware whether they're mosaic or not. So those are the types of things I think it's important for breeders to know. Next slide, please. Okay, so I use this little graphic because to me it kind of encapsulates what I've said, and that is you can have an ice cream sundae, and that is what our basic genetic improvement programs are. And they start off with an association of like-minded breeders, that's your breed associations, who get together and say, what's our breeding objectives? What do we want our breed to do? Do we want it to be maternal? Is it going to be terminal? What's our characteristics? Then you're going to have breeders recording performance information using AI, ET, genomic selection, maybe even cloning. And then you could have a cherry on top of your ice cream sundae, which would be genes that have been introduced using genome editing. It builds on the already elite genotypes. It's not a technology that works on its own. You have to incorporate it with our current breeding and selection programs. That's where its value is, not as a replacement. So I argue it's a synergistic, but not competitive with conventional breeding programs. Next slide, please. So in summary, you can use genome editing to introduce useful genetic variation with alleles such as disease resistance and welfare traits like polled without crossbreeding. Next. Scaling it to commercial livestock situations is not for the faint of heart. And if you have a look at what Genus has done with their PRRS-resistant pigs, you can have a look, they're a publicly traded company, at the hundreds of millions of dollars they're spending introducing it into all of their paternal or grandparent lines of the pig breeding pyramid to get all both the maternal and the paternal lines homozygous, such that that will then pass down to the commercial livestock. So you need to edit multiple founders, as we would in cattle. You don't want to do one bull. That's going to narrow your genetic base too much. It's going to have to be widely used in that regard. Regulators in many countries are considering simple edits like knockouts with no foreign DNA to be no different to conventional breeding. That's not the approach that's being taken here. They're all being treated as unapproved animal drugs. Just sometimes you can sell your unapproved animal drug and they won't exercise their enforcement on it. What that does for trade, when the EU asks if there's any meat from gene-edited animals in our beef supply chain, I don't even want to think about it. So hopefully they won't ask. The fate of genome editing will depend upon developing sensible regulations, risk-based. At the end of the day, if you're doing a knockout, it's literally no different to the millions of knockouts that exist in cattle right now. That's what your breeders all work on. And so to me, there's not a bright line in the sand that says, just because I did it, it's somehow more dangerous than when it's done naturally. That doesn't make a lot of sense from a scientific perspective. And really, the trade aspect is going to be super important. And I know Australia has a similar approach to South America, but the large pastoral organisations there are very nervous to introduce polled, even though it's going to be mandated, because they are worried about consumer pushback with especially their Wagyu branded products going into some of the Asian markets and what the consumers are going to do there. And so we have this interesting kind of, I don't know, trade-off or war between, do we want animal welfare or do we hate GMOs more? Which one do we want to deal with? And so it's a very powerful technology, but it also comes with a lot of regulatory and potential public perception baggage, I guess, that potentially will make it less useful than perhaps it's going to be in other countries. And I think that's my last slide. If you, click. Oh, I'm organising the World Congress of Genetics applied to livestock production. It's every 4 years around the world, and Wisconsin is hosting it in 2026. So if you really want to get into the quantitative genetics and very scientific presentations, that would be a good conference to go to. So with that, thank you very much.